Dear students, in this screencast lecture, we are going to learn about the enzymes involved there in the nitrogen assimilation, that is taking nitrogen inside, and how that whole process is getting regulated. The nitrogen assimilation is a process by which the inorganic forms of nitrogen such as nitrogen gas, nitrate or ammonia are all converted into organic forms, which includes amino acids and nucleotides. These organic forms will in turn leads to the formation of the other macromolecules such as proteins and DNA that are essential for the functioning of the cell. This process is essential for a cellular metabolism and it is catalyzed by several specific enzymes. So this assimilation takes place mainly in the bacteria and even in the plant systems. As animals are consumed the preformed organic foods, the proteins or amino acids will be directly assimilated instead of assimilation from the inorganic forms. Here we look at the list of enzymes that are involved in this process. The first one is a nitrate reductase and the next enzyme is a nitrite reductase. The function of nitrate reductase is to convert the nitrate ion into nitrite. This process takes place there in the cytoplasm of the cell. If you look at the reaction, nitrate in the presence of NADPH is converted into nitrite and NADP and water. Then we look at how this enzyme is regulated. The regulation of this enzyme is mainly mediated by the nitrate availability, and the enzyme function can be stopped or repressed by the presence of ammonia there in the system. This kind of a repression is commonly referred as a feedback inhibition system operating in an organism. It can also be regulated by the phosphorylation and dephosphorylation, that is a degradation of the phosphorylated form. The next enzyme important there in the nitrate reduction is the nitrite reductase. Its main function is to convert the nitrite into ammonium ions. These ammonium ions are those that can be incorporated directly inside the cell. This process will be commonly taking place there in the plastids or chloroplasts of the cell. The reaction involves nitrite in the presence of ferredoxin in the reduced form is converted into ammonia, and ferredoxin is oxidized into ferredoxin of oxidized form, and finally the water molecule will be formed. If you look at the regulation of this enzyme, it is mainly get activated with the presence of light, especially there in the photosynthetic organism. Apart from that, based on the demand of nitrogen by the cell, this particular enzyme is regulated. Next, we look at how the nitrogen that have been fixed by the prokaryotic organism, that is nitrogen is converted into ammonia, and how it gets assimilated there inside the cell. The below mechanisms were specifically pertaining to microorganisms that are all capable of fixing the nitrogen, that is technically they are referred as a diazotrophs. These organisms have the ability to convert atmospheric nitrogen into ammonia. This ammonia is getting incorporated there into the amino acids, which are in turn help in the formation of the proteins. The first enzyme involved here is a glutamine synthetase. Its main function is to incorporate the ammonium into the glutamate, that is the amino acid, to form into the glutamine. How this enzyme is regulated? It is again mainly by the feedback inhibition that could be initiated there by the glutamine, that is the final product of this enzyme. Apart from that, post-translational modification that could be resulted mainly by the adenylation there in the prokaryotic cells could also able to regulate this enzyme function. The next important regulation is the control of the carbon to nitrogen balance inside the cell. The next enzyme is a glutamate synthase, which is also referred as a GOGAT or glutamine 2-oxoglutarate aminotransferase enzyme. Its main function is to transfer the nitrogen from the glutamine to a alpha ketoglutarate molecule to form into 2-glutamate molecule. The reaction has been shown there. Next, we look at how it is regulated. It is co-regulated along with the glutamine synthetase enzyme, which we have seen earlier, and it is also sensitive for the cellular energy levels, mainly the NADH available in the cells. And the next important enzyme is the glutamate dehydrogenase enzyme. The way in which the nitrogen getting assimilated using this particular enzyme is referred as an alternative pathway. then it means that glutamine synthetase as well as glutamate synthase based assimilation is the primary pathway of nitrogen assimilation. This enzyme incorporates ammonium ions there into the alpha ketoglutarate glutarate molecule with the help of energy in the form of a reduced equivalent, that is NADPH, that results in the formation of the glutamate. Then we look at how this enzyme is getting regulated. Functioning of this enzyme is mainly under a nitrogen excess conditions, and it is regulated by the carbon to nitrogen balance that have been available there in the cells compared to the other enzymes, mainly the glutamate synthase as well as glutamine synthetase. This enzyme does not play a much significant role there in the organism. That's the reason it is referred as an alternate pathway by which nitrogen is getting assimilated there into the cells.